Hi, I'm Jessica Petersen, an associate professor of animal genomics at the University of Nebraska-Lincoln, and I'm excited to present to you today the work that we've done regarding a new old coat color in Dexter cattle. We've called this the Carol Davidson project. So I'm going to start out by acknowledging Carol, because this would not be happening at all if it were not for her. Carol emailed me almost 2 years ago to the day, and I hadn't met her before. She didn't know me. Somebody gave her my information and said I might be able to help her with a question she had regarding a unique coat color in Dexter cattle. So after we had corresponded, Carol said she was stalking me and looking at what I looked like in my pictures online. And so she sent me pictures of what she looked like. And so this middle picture is one she sent from a vacation when she was in Egypt. And here she is over here in 1997. I believe it was her bull that sired that calf that was a champion calf. So one thing Carol said in her email that really made me want to be involved in this more was she said that she acknowledged this wasn't a groundbreaking question, but she considered herself homozygous for the curiosity gene. So she just wanted to have an answer of why these animals were colors that maybe they shouldn't be. I also want to make some acknowledgments. So the American Dexter Cattle Association and Wakarusa Ridge Ranch, along with the Carol Davidson estate, financially supported this project in honor of Carol. And genetics takes some money, so we really appreciate that support. I also want to acknowledge, and I will many times throughout the presentation, the breeders, Dexter breeders worldwide. And I'm going to name three that were particularly important in this research. So Patty Adams and Wakarusa Ridge Ranch, Nico and Annemiek at Pork Prunus Farm in the Netherlands, and Louise Ball at the Carrigain Dexter Stud in Australia. They both sent quite a few samples. They all sent quite a few samples that really helped us move forward. And then when we put the call out for other samples, there were 20 other breeders and farms that sent samples. We had 200, I believe, 17 samples that were included in this project. And that's thanks to everybody here. In addition, I want to acknowledge Carol's friends and family, so Michelle and Owen, who helped get all of the materials that Carol so carefully put together to us and the information so we could do this work. And then while I'm talking about this work was performed by Anna Fuller, who is my lab manager and technologist. So she was the one actually in the lab that was isolating the DNA, looking at the sequences and doing all the comparisons. So this was Anna's project. Then I will also say that I found in Carol's notes, and she put together an amazing binder of information with pedigrees and pictures and other publications. She had a page of acknowledgments, so I want to share those. So she acknowledged several different people who got her interested in this project and who helped keep her going, as she said, along the way. Some of those names I've already said, but they also include Veronica Schofield, Duncan McIntyre, and John O'Neill, who we spoke with and who helped us along the way as well. Nico again, Di Smith, Kathy Heinzelman, Nikki Moran, Carol named herself there, and then John Potter. So since Carol thought these people were important, and I know they were, I wanted to make sure they got acknowledged as well. So what was this question that Carol had? And so what Carol and others had noticed was that there were some Dexter cattle, which we would call done, that if you genotype them, they weren't done. They didn't have the correct genotype, the little b that's necessary to be what's considered a done. So this animal's big B, it should be black. Similarly, there are cattle that looked cream or blonde colored that also had the big B, and in fact, this animal was genetically red. So it should be red and not that cream color. So the question was, what causes that? And as we went along with this project, we started calling the darker colored ones chocolate and the lighter colored ones cream. So for better or for worse, that's what I'm going to call them through this presentation. Okay, so we're all here because we're interested in coat color to some extent, but I'm going to do a little bit of a review because it's important that we understand that the basics are on the same page so I can explain this new variant that we found. So in most every animal, it comes down to the first question of, is the animal red or is it black? So we're going to start with red. Red Dexter cattle can have any combination of the genotype E plus or little e. That E plus is often called wild red. If the animal has that, especially the males, might have a darker color around their head and their legs. But there are a lot of variations of red. It can become really shiny copper color to a cherry red, and everything in between. I'm going to take a little side tangent for a second, because this is something we didn't expect to find, but we did find in this study, is that Dexters also have a different variation of red, and that's called EV1. This was described initially in some cattle in Sweden. I want you to think of it as basically E plus, as a variation of wild red, because it's a mutation that is on that wild red background. So it probably acts pretty similarly. We didn't have a large enough sample to ask the question of, are those animals different? But they all are red. They have that base color red, and so we want to consider them all the same. But we did find several animals, multiple animals, who had at least one and sometimes two copies of the EV1 variant. To my knowledge, that's not specifically genotyped for in any genotyping labs. If I were to send hair samples from this animal, for instance, to UC Davis, it would come back as E plus E plus. That's demonstrating that relationship of those two different varieties of red to one another. So after my tangent, let's go back to the other basic coat color, which is black. Black is dominant to red, so if your animal has one or two ED alleles, it's going to be black. So this animal is homozygous for black, where these three have one copy of ED that makes them black, and then they can have anything else with that. So in this instance, these animals each have one copy of a different variety of the red allele. So we often say that those animals are black, but they carry red because they can give that red to their offspring, and they can have a red-colored offspring depending upon to whom they are bred. So we have red and black, but we're not here to talk about that. So let's review first the first dun that was discovered, dun or brown. So it's a mutation in the TYRP1 gene. It was published in 2003, and you've been able to do genetic testing for it. What this does is that it takes a black animal, so an animal that has D, the ED, and if it has this little b genotype at this mutation, so it has to be homozygous, it dilutes that black color to any of these different shades of dun. And just like there's different shades of red, there's many different shades of dun, from this more blonde looking cow here to this really kind of brown looking bull and everything in between. This is the dun that is known, but it does take two copies of the little b to be dun. So for instance, you can have a black animal that only has one little b, but he has one big B. He's not going to be dun in color. He's going to stay black, or she. So this animal we say is black, but carries dun because it has one copy of that little b. So red, it's a little bit different, but red can also carry dun. So if this animal is E plus, or whatever, and has one little b, we could say this animal carries dun. But I mentioned at the beginning, the dun mutation only affects black coat color. So even if this red animal were to have a little b genotype, it's not going to show up as dun. It's going to be a red animal. So I say this is red and it's hidden dun, if you will, because it actually has two copies of the little b, the dun locus. So remember that Carol's quest was to figure out what caused these animals that look like they maybe should be dun to be the colors they are when they don't have the little b genotype. So this quest that we were given is to figure out what causes that. And from pedigree records and what Carol had put together and what we were seeing too, it appeared that these are probably the same thing, but the difference is this animal is otherwise black, while this animal is otherwise red. So they have something that's diluting both the black or the red to be a lighter color. So what did we do? We sequenced the whole genome, so all 2.6 billion ACTs and Gs of the genome of 5 different Dexter cattle. 3 of them were chocolate, 1 of them was cream, and 1 of them was a calf that was black from one of the chocolate dams. So if what we're hypothesizing is that if it has to be homozygous, that these 4 animals will have 2 copies of whatever this new thing is. This animal will only have 1 copy because it's black, so it's not dilute, but it came from a chocolate animal, so it had to inherit one of them, one of those alleles from that chocolate dam. So we then put all that information into a computer, and it does some statistics, and it filters out only the variants in the genome. So it narrows it down to only the parts of the genome that fit that hypothesis, where these 4 animals share homozygosity for something. This one is carrier of it. And then we further said, well, let's see. We don't think that cattle of other breeds, of other major beef breeds, probably have this. Dexters aren't usually bred in with those other animals. And furthermore, we had animals that we knew did not have the dilute coat color. So we could rule out things that were in all these 5,000 other cattle that might be shared with our chocolate and cream animals. Unfortunately, we have a good collaborator at the University of Missouri, Dr. Robert Schnabel, who allows us access to his database of over 5,000 sequences of different cattle. So we're here because we found it. So we found a mutation in the gene SLC45A2. It's not, like I said, it's not a fun name or an easy to say name, but that's the name of the gene. So all the chocolate animals we sequenced were black. They had the ED, and they had this genotype CC at this gene, at this position in this gene that we found. The cream animal was otherwise red. I believe the one that we sequenced was E plus little e, and CC at this new position in this gene SLC45A2. But that's not quite enough to say, oh, that has to be it, right? So we went to samples that you all sent us. And we had 63 animals that we would probably call done. And they had the ED, so they were a black animal that looks dilute. Sure enough, 55 of them had the little b genotype for done. So these were our the traditional, if you will, done Dexter cattle. That leaves 8 of them that didn't. And all 8 of those we could explain by the CC genotype at this SLC45A2 gene. So they all had this, were homozygous for this new variant that we identified. Same thing was the case for what we're calling the cream cattle. So this little guy did not have ED. It should have been red, and it didn't have little b, even though if it did, it probably shouldn't have an effect, right? Because the little b doesn't affect red animals. But all of those 6 cream-colored animals were CC for that SLC45A2 allele. Okay, so we found that it can explain the dilute color of animals that are not, that look done or cream, that do not have the TYRP1 mutation. And we also found in the 227 samples that we genotyped that none of the red or black Dexters had the CC genotype. So this also helps demonstrate that this is causative of the dilute color and not of the red, and it doesn't affect, I'm sorry, that any animal that is red or black doesn't have that genotype. So when we find something like this, our next question is like, are we really sure? Does that gene make sense? Do we know that gene has an effect on pigment production or in hair color? And it does. So we study horses in my lab as well. We didn't identify these mutations, but there's different mutation in the same gene, SLC45A2, that acts very similarly in horses. And in horses, it's called the Pearl mutation. So in horses that are homozygous for this mutation in that same gene, makes a black horse look like this kind of chocolatey brown color called Pearl. And it makes a red horse look cream colored or blonde. So this is more confirmation that, yeah, it probably makes sense that this mutation we found is doing what we think it is, and it's diluting both either the black color of Dexter's to the chocolate or the red color to the cream. And I think this is just fun because almost all mammals share the same sets of genes. We just have different variations of them. And so there's different mutations in this exact same gene that has been found in many, many animals, including white tigers. That explains the pigment dilution in white tigers. The white gorilla. Some true albino animals are attributed to mutations in this gene. And the only other mutation in this gene we could find in cattle is in Bronve cattle. There are two calves that had mutations in this gene, and they were albinos, so they had lack of eye pigment as well as hair pigment. So the next question I'm sure somebody might ask is, where did this mutation come from? So we often like to think about what bloodlines, where did this originate? And when we do studies in the lab, one of the things that we always are careful about is confidentiality in animals. We don't want bloodlines to start to be talked about if maybe the mutation isn't identified yet or anything like that. That's not our place. Our place is to identify them. In this case, however, I have permission from the person who owns the sample from this animal to talk about it. And in fact, there are several people that were contributed to the study who had these chocolate or cream animals that said, we've noticed that there's one bull in the pedigree of all those animals. And so we were able to genotype from semen of this bull and confirm that Shadwell Robert was indeed a carrier of our chocolate and cream mutation. So his genotype was EDE+, he was big B at the Dunn locus, and he was NC, so a carrier of our newly identified chocolate and cream mutation. It's newly identified, but it's not new. Carroll's notes actually said that it was first observed as far back as 1889, I believe. So that's old. At that time, she reported that people didn't want it, and so they bred it out of the population. But it was a few years ago when it started showing up again more frequently, which is when she brought this question forward. So Shadwell Robert, I'm almost certain, did not originate the mutation, but he was probably the main or one of the main perpetuators, probably one of the more popular sires recently that has had it, that's been used to make it become more common in the population. There's things we still don't know. So for instance, we haven't identified any cattle that are both little b, so that should be done, and CC, that are chocolate or cream. The genes are totally separate, so there should be a possibility that you could have animals that have both chocolate and dun. We have no reason to suspect that it causes anything detrimental, although a question was asked, could it be lethal if they're homozygous for both? We just don't know the answer to that. I think more likely we haven't seen animals who have both just because they are in a little bit different bloodlines. And the C allele for the chocolate cream is still quite rare. So I think it's more of a issue of statistics and chance that we haven't had them all come together yet to see what happens. One might hypothesize that an animal that has both dun and cream is going to be even lighter in color, but that's yet to be seen. People who have done cattle, as well as chocolate or cream cattle, have also noted that it alters the eye pigment, and that's consistent with mutations in this gene in other animals, especially those albino animals. They clearly have very light eye pigment. This is the case in chocolate and cream animals, but we really haven't studied, and nobody's studied to my knowledge, if there's different variation in how that eye color is affected, or if it might affect the function of that eye. But I don't have any reason to think it does from people who have had these animals. There's been no reports that I know of that say there's anything bad about it, except they just have these very striking lighter colored eyes. So in summary, there's two ways a Dexter can have dilute color. We now know both of those ways. Either the dun, little b, which dilutes the black coat color, or what we're calling chocolate and cream cattle, which is the CC genotype at the gene SLC45A2. So the difference is that dun does not change the color of red cattle, but the chocolate and cream mutation does. So if you have a dun Dexter or a Dexter that you said, well, that looks dun, but it doesn't test to be done with the TYRP1 locus, it has the chocolate or cream mutation. So it might be a good idea to test to see if that's the case. And testing is going to be available. UC Davis has this ready to go. When I spoke with them last week, they said it should be released at the end of August. And when that's the case, I'll let the association know and make sure it gets posted on the social medias and things like that. But this is what the report's going to look like that you will get. 2 alleles, N is for normal and C is for dilution. I'll take a little side note. Originally, the UC Davis folks suggested it'd be D for dilution. And I said, well, C is kind of important to us for three reasons. One, we're chocolate and cream. Those are C's. The actual nucleotide of DNA is a C. And then we were trying, when we started this project, how could we name this after Carol? So we're using C to stand for all those things: chocolate, cream, the C nucleotide, and also for Carol. So when you get your results, you'll see that your animal will either be NN, so that means it doesn't have any copies of it. It's not going to be a dilute unless it has done. Your cattle can be a carrier of the chocolate cream allele, so it'll be NC. They won't have any diluted pigment in their coats, but they could pass it on to their offspring. And I should say they could have diluted pigment due to done, but they don't have diluted pigment due to this mutation. And then cattle with CC, whether they're black or red, will have diluted pigment in their coats, and they will pass that on to their offspring 100% of the time. So like I said, this should be available at the end of August at UC Davis. So finally, I acknowledge a lot of people in the beginning, and I want to acknowledge now that the people that work in my lab. So not only Anna, but I get to work with a lot of talented students. We work on a lot of different projects in cattle and horses primarily. These are the people in the lab actually doing the work. So I'm fortunate to work with a great group of people. And at this point, I'll take any questions.